Okay, guys, today we're going to look at something called epistasis, and this occurs when the expression of one gene is influenced or affected by the inheritance of one or multiple other genes. And the two examples we're going to look at, the two traits we're going to look at, are eye color and hair color. So think back to the beginning of the unit. We mentioned eye color, and I had told you that brown eyes are dominant to blue eyes. So we can see here that depending on which allele somebody inherits from each parent is going to contribute to their genotype, which then influences phenotype. So brown eyes being dominant, you could be homozygous for brown eyes or heterozygous, and homozygous recessive would result in the blue-eyed recessive phenotype. There's a problem with this, though. There are many other shades of eye color besides brown and blue. For example, what about green eyes? People have green eyes. So we're going to look at how eye color is actually inherited and see that it's much more complicated than just one gene and this one dominant and recessive allele. We're going to use this model here. And this is looking at two different genes that actually contribute to eye color. Again, like all things that we've been going through, it's actually a little bit more complicated than this, but we're going to stick to two genes to make things not too hard to visualize. So we're looking at these two genes here. Gene number 1 is located on chromosome 15, and then gene number 2 is located on a completely different chromosome, chromosome number 19. For gene number 1, we're using letter B's, so capital B and lowercase b. And then for gene number 2, we are using, for some reason, we are using capital G's, and instead of lowercase g's, we're using a lowercase b, which can be a little bit confusing. So I just want to point it out here because that's not typically how we do things. But this is how this chart was made. We're going to go with this. So some rules when looking at this chart, because I don't expect you to memorize these genotypes and phenotypes, I expect you to look back at this chart and use it, is that the big B is going to be dominant to big G. So the big B is going to mask any appearance of the big G or the little b. The big G is going to be dominant to the little b, and that little b is the most recessive allele. So you'll see that in the blue phenotype, the only genotype that results in that would be being homozygous recessive for both of these genes. So little b, little b, little b, little b. Let's apply this to some of our genetics problems. So we're going to go back for a second and look and remind ourselves of how we actually figure out mom and dad's gametes. Since we're looking at two genes now, we need to approach our Punnett square a little bit differently. So I want to remind you how meiosis and figuring out those possible gametes for mom and dad, how that relates to the Punnett square. Something we need to talk about is independent assortment. Since the two genes for eye color are carried on non-homologous chromosomes, so remember I said gene 1 is on chromosome 15, gene 2 is on a completely different chromosome that has other kinds of genes on it, is on chromosome 19. So since they are on non-homologous chromosomes, they show what's called independent assortment during meiosis. Independent assortment, in addition to crossing over we had talked about, also contributes to such genetic variation within a parent's gametes, which leads to why siblings can look so different from each other. So we're going to start with mom here in our problem. Dad is big B, big B, big G, big G. And mom is big B, little b, big G, big B. So mom's genotype's a little more complicated for figuring out possible gamete combinations. So we're going to look at hers first. And keep in mind, you can go back and use this chart to figure out what mom and dad's actual eye color are. I don't expect you to memorize these. Go back and look at the chart. Okay, so during meiosis, remember, homologous pairs line up during meiosis, and then those homologous pairs split during meiosis 1, and then those sister chromatids split during meiosis 2, resulting in those 4 haploid gametes that have half the number of chromosomes. So mom's genotype is big B, little b, and we can see that gene represented here in the chromosomes that are a little bit larger. So she inherited a big B from one parent and a little b from another parent. And then she is also, for the second gene, big G, little b, And we can see that that gene is on a completely different chromosome. We can see that since it's smaller in size here. So she inherited a big G from one parent and a little b from another parent. Independent assortment refers to the fact that since these genes are on completely different chromosomes, they could line up this way, right? Just like we have them, then these homologous pairs would separate, and that affects what ends up in the gametes here. So if we're looking at this, when these homologous pairs separate, and then those sister chromatids separate, we would end up with gametes that have, so our first set of gametes with the blue chromosomes in them would have a big B, big G. I'm going to fill that in here. Big B, big G. And our second pair of gametes here, so 50% of the gametes, half of the gametes would have little b, little b. But since those genes are on different chromosomes, the chromosomes might not line up that way. This is why we say that they sort independently. That's independent assortment. So the chromosomes could line up in this way. Notice here we have the blue, red, and that's just representing the different chromosomes that one came from mom, one came from dad. And here we have the red, blue. So we switched the order here. And that's going to affect what alleles end up in mom's gametes if the chromosomes happen to line up this way, those homologous pairs. And again, it's random. Could happen this way, could happen this way. We need to account for both because we don't know exactly what happened when mom's gametes were being formed. Okay, so if we look at our second option here, and these homologous pairs split, and then those sister chromatids split, our gametes could also end up with the big B, little b combination, or the little b, big G combination. Okay. So that is how we can figure out that mom actually has 4 possibilities for her gametes because of independent assortment. Now, when we look at dad's a little bit easier to figure out, right? Because he is all homozygous dominant, he can only pass on a big B and he can only pass on a big G. So his only possible combination for gametes would be big B, big G. Now that I showed you that, I want to actually show you that we don't have to draw out meiosis. There's a simpler way to figure out gamete combinations when we're looking at multiple genes controlling a trait. And it's actually going to sound, hopefully, very similar to something that you do in your math class. And we call this the FOIL method. So I showed you meiosis to explain where we're getting those combinations from and why they can be so different because of independent assortment. But I don't expect you to draw out meiosis each time. You can use this FOIL method when you're trying to figure out gametes for mom and dad. So given that, we're going to start with mom again because she's a little more complicated. Given that we said mom is big B, little b, big G, little b, we're going to use this FOIL method to figure out her 4 possible gametes. Okay, so FOIL stands for first. So we're going to pick the letters that are first in our genotype here. So the first in the gene pair. So that would be this B. And this big G. So they're the first in each gene pair. So the first 2 letters are representing the first gene, the second 2 letters are representing the second gene, the first allele in each of those gene pairs. So our first option would be big B, big G. And then our next letter, O, represents the outside. So the outside would refer to this big B, and then this little b. So that's our second combination, big B, little b. And then the I in FOIL is referring to the inside. So we're going to go with this little b is on the inside and the big G is on the inside. And then the L in FOIL is referring to the last allele in each gene pair. So that would be the little b here and then the little b here. And you will notice that using this FOIL method, I was able to achieve the same exact combinations as I did working through the meiosis that could have occurred. Now with that, we can FOIL also. So we're going to start again with the first. Let me move my little lines over here. All right, so the first in each gene pair would be big B, big G. The outside would be this big B and this big G. So remember I said, because Dad's homozygous dominant for both, he's pretty easy to figure out. There's not too many options here. The inside would refer to this big B and this big G. And the last would refer to this big B and the last allele here, big G. So Dad really only has one option for his gametes, right? So we could fill Mom in on the top, her 4 possibilities, and we just need one row since Dad can only give a big B, big G. So you should be able to now use that information to fill in this Punnett square here. All right, so we have Mom's 4 possibilities, we have Dad's only possibility, and then you should be able to fill in the Punnett square and answer the questions. Remember, when you're trying to refer the genotype, look at the chart. I don't expect you to remember genotypes and phenotypes.